Hello viewers and welcome to biology class. I'm your teacher Wamaya Kevin and you're on the continuation of cell physiology. Topic is still cell physiology. Cell physiology. So we defined the cell physiology and physiological process. There were three. We are already through with the diffusion. So today we go straight to osmosis. 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 So what is osmosis? We go straight to the definition of osmosis. I have two definitions for osmosis. So number one is the process by which solvent material materials move from a region of high concentration to high concentration that is dilute into bracket dilute solution into a region of low concentration into bracket we say highly concentrated solution, concentrated solution across semi-permeable membrane. So we are being told that osmosis is the process by which solvent water molecules or solvent materials move from a region of high concentrated, where they are highly concentrated, that is what? Dilute solution, a solution that has less solute, to a region of lowly concentrated, that is the solution that is what? Highly concentrated across semi-permeable membrane. And say a cell membrane is one of the factors or one of the characteristics of a cell membrane was that it was semi-permeable. We say semi-permeable, it's able to enable small water molecules to pass through. Another definition of osmosis, or we say a special type of diffusion, a special type of diffusion since it involves movement of solvent into bracket water. Example of solvent is water molecules into bracket water molecules from a region of high concentration to a region of low concentration. So in short, for osmosis to take place, we must have the two type of what? Solution, whereby one solution has to be highly concentrated and the other solution has to be lowly concentrated. So water molecules are going to move from where they are highly concentrated to a region where they are lowly concentrated across semi-permeable membrane. Semi-permeable membrane is one of the properties of a cell, and we can also use artificial cell membrane. We use a whisking tubing. So we have two definitions for osmosis: the process by which solvent materials move from a region of high concentration, that is a dilute solution, into a region of low concentration, that is a highly concentrated solution, across semi-permeable membrane. Or a special type of diffusion, since it involves movement of solvent, that is water, molecules from a region of high concentrated to a region of low concentrated. That means we are having two types of solvents. One, we take just distilled water, and number two, we take water, then we add what? A solute. You can either add salt or sugar to make it highly concentrated. So you will see that the water molecules will be moving from distilled water into salty water. That's how osmosis does what works. So we go to practical demonstration of what? Osmosis. Practical demonstration. Practical demonstration of osmosis. So, we want to see how osmosis works on what? A living cell. So in this case, we want to start with the first cell membrane. We are going to use artificial one. And artificial one, I said, we are going to use a whisking tubing. So in our beaker, there is whisking tubing. And inside the whisking tubing, we add sugar. It is having sugar solution. And outside the beaker and from the surrounding, it is what? Distilled water. Distilled water. So inside the whisking tubing, we have salty or sugar solution, and outside we have distilled water. So the water is going to move from the beaker into the whisking tubing across semi-permeable membrane. What is the semi-permeable membrane here? It is what? The whisking tubing. So at the end of the experiment, we are going to have such a diagram. So after 30 minutes, the whisking tubing will swell up. It's going to swell. And the water inside the beaker will do what? Will drop. So this is distilled water, and this is sugar solution. So at the end of the experiment, A, and at the beginning of the experiment, we mark it as A, and at the end of the experiment, we mark it as B. So if you look at the board, you can see, you'll be able to see that the water level in the beaker has dropped. But here, the water level in the beaker, it is what? Full. And the solution inside the whisking tubing, which is a highly concentrated solution, has reduced. But the solution inside the whisking tubing at the end of the experiment, it has increased. The volume of the solution has done what? Increased. That means that water moved from the beaker into the whisking tubing. It moved from where it was the water molecules. The water molecules inside the beaker are high, while the water molecules inside the whisking tubing in the sugar solution are what? Low. So the water moved from a highly concentrated region to a lowly concentrated region across semi-permeable membrane. And what is our semi-permeable membrane here? Is the what? Is the whisking tubing. So explanation of that you can say, explanation. Explanation, you say, whisking tubing becomes swollen. Whisking tubing becomes swollen, indicating that its cell's content has increased, indicating that its cell content has increased. Another point, the amount of water in the beaker has decreased or reduced, has decreased. The amount of water in the beaker has decreased. This implies that water moved from the beaker into the whisking tubing by osmosis. This implies that water moved from the beaker into the whisking tubing by osmosis. By osmosis. So these are our explanation. Explanation here indicates or shows what has taken place here. That whisking tubing becomes swollen, indicating that its cell content has what? Increased. Definitely you can see here the content of the whisking tubing did what? Increased. The amount of water in the beaker has decreased. Here the beaker is filled up, but here the amount of water has reduced. Why? The water, we're being told, the amount of water in the beaker has decreased. This implies that the water moved from the beaker into the whisking tubing by osmosis. Another one this one, I've just demonstrated osmosis using what? Artificial cell membrane, that is a whisking tubing. What about if we use a live cell membrane? So this practical one, demonstration one. Number two, we can use this one, whereby we take this our beaker again. This is our beaker. And inside our beaker, we place water. Then on top of it, we place Irish potato. Irish potato. Inside the whisking tubing, we place our Irish potato. Inside the beaker, we place our Irish potato. So this one should be our Irish potato. So here, we mark it as what? Irish potato. And here is what? Water. That is distilled. Distilled water. What's going to happen is that the content inside the cell, the content inside the cell of Irish potato is highly concentrated than the surrounding water in the beaker. That means that water inside the beaker is highly concentrated. It's having a lot of what? Water molecules than the cell cytoplasm. So the content of the cell cytoplasm has less solvent than what? The beaker. Now, what is going to move from what? From the beaker into the cell of Irish potato, and it will become what? It will swell, and it becomes what? Turgid. So explanation, and explanation you say, under explanation you say, water in the beaker has more water molecules than the cell cytoplasm. When we talk about the cell cytoplasm, is the cytoplasm of what? Irish potato. Next point is say the water will move from the beaker into the cell by osmosis. Then next point you say water will move from the beaker into the cell by osmosis. Next point to say that the cell will swell and becomes turgid. and becomes turgid or tough. That's the two practical demonstration of what? Osmosis. We can either use a whisking tubing or what? An Irish potato to demonstrate what? The osmosis. So another explanation, we continue the explanation. So up to this point, you should be able to know what is osmosis, definition of osmosis. Number two, you should be able to know the two types of concentration. Is water molecules moving from where they are highly concentrated region to a lowly or the vice versa? It should be from where they are highly concentrated to where they are lowly concentrated. And I say when the solution is low concentrated, it's having what? When the solution is highly concentrated, it means that it has less solvent and more solute. But when the solution is highly concentrated, like when you say it is hypotonic or hypertonic, hypotonic has more solvent than the solute, but hypertonic has more solute than solvent. That's the differences. So you have explanation on that. We say, when two separate solutions are separated by semi-permeable membrane, when two separate solutions are separated by a semi-permeable membrane, water will move from a region of highly concentrated, that is from a dilute concentrated solution, to where there is what? Highly concentrated solution across semi-permeable membrane. So you write, a semi-permeable membrane, a semi-permeable membrane does not allow, a semi-permeable membrane does not allow movement of solute particles across it. Not allow movement of solute particles to pass across it. That means that when it's on the diagram that I've just drew, we were able to see that solute, that is in the whisking tubing, we had what? Sugar solution. And the content of the sugar solution were unable to move from the whisking tubing into the beaker. That's why, because it had what? Solute particles. Only water molecules are able to move from the beaker into the whisking tubing. That's why it is. That's why it says what? It is semi-permeable, or partially permeable, or selectively permeable. Next point we write. Movement of water molecules continues until movement of water molecules continues until it reaches equilibrium. Equilibrium, where the two solutions have the same concentration. Where the two solutions have the same concentration. And the solution is said to be, full stop, and the solution is said to be isotonic solution. Here means that when water that is highly concentrated, like when we place it in the beaker, where in the beaker the whisking tubing was highly concentrated, and in the beaker was what? Lowly concentrated, whereby in the beaker, the water was what? Hypotonic. That is distilled water. And in the whisking tubing, the solution was what? Sugary. So you see, in the whisking tubing, absorbed water, or the water was able to move from the beaker into the whisking tubing, up to a point where now the whisking tubing swelled up. After swelling up, the water level inside the beaker dropped up to an equilibrium, where now the content of the whisking tubing and the content of the beaker were the same. The solution inside the whisking tubing and the solution inside the beaker were the same. That's why we say that the solution now is what? Isotonic, and osmosis cannot take place again. That is, the osmosis has reached what? Maximum. It cannot take place again. Why? Because the solutions are the same. We call it isotonic solution. Next point, a highly concentrated solution is called hypertonic solution. Is called hypertonic solution because it has more solutes and less solvent. That means that it has more solutes than what? Solvent. And in chemistry, we're told that solute, it is in solid form, e.g., we have salt. And solvent is water. So when solute and solvent mix, we get what? A solution. So in hypertonic solution is highly concentrated solution. It has more solute than what? A solvent. And we have a lowly concentrated solution is called hypotonic solution because it has more solvent than solute. Because it has more solvent than solute. That means that the only difference is O here. Here we have O and here we have E. This is called hypertonic and this one is called hypotonic. Hyper for highly concentrated, hypo for lowly concentrated. And the difference is that in hyper, we have more solute than solvent. So we have a lot of what? Solute molecules or particles and less solvent molecules. But in hypotonic, we have more water molecules and less solute molecules. Next, we move to the osmotic pressure as a subtopic. Osmotic pressure. Osmotic pressure. Now, definitions, go straight to the definition of what? Osmotic pressure refers to the force by which a concentrated solution draws water to itself. When a concentrated solution is separated, remember a concentrated solution I said is a solution that has more solute than solvent. Its example of the name is called what? Hypertonic, a highly concentrated solution. So I'm being told, when a concentrated solution is separated from distilled water by a semi-permeable membrane, the concentrated solution will develop a force which draws water. So if I have a demonstration here, I say we take it like this, and here I partition into two. My beaker I partition into whereby this side it is low concentrated and this side is what? Highly concentrated. What's going to happen that when you place a highly concentrated region and a lowly concentrated region, you're going to find out that water molecules will have to move from one side to another. Which one do you think is going to what's going to happen? You'll find out that water, this side, let me say that the level of water, is this the level of water at the beginning of what? Experiment. And this is what? A cell. This a plant, an animal cell. Let us say it's an red blood cells, and you place it what? In a lowly concentrated region. What's going to happen is that water from this side, water will move from this side to this side. From where it is low to where it is what? High. And the water content here is going to do what? To drop at the end of what? Experiment. The water in the lowly concentrated region is going to drop. Why? Because water will move from where it's less to where it is more. Then you're being told when a solution is separated from, when a highly concentrated solution is separated from low concentrated solution by a cell membrane, it will develop a force. It will develop a force which draws water through. Which it draws water through. Which it draws water through a semi-permeable membrane. Through a semi-permeable membrane. Through a semi-permeable membrane. At the other point of being told, the distilled water, which is highly concentrated, is going to move from where it is highly concentrated into where it is what? Lowly concentrated. And how do we measure osmotic pressure? We have an instrument called an osmometer. It's an instrument used to measure osmotic pressure. An osmometer is an instrument used to measure osmotic pressure. Now, you place a cell. We are having two cells. Having one cell that is placed in what? A highly concentrated solution, another one is placed in what? A lowly concentrated solution. In this regard, we are being told we have two types of what? Solution. Concentrated and low concentrated. So when you place, they have said here, when a highly concentrated solution is separated from a lowly concentrated solution by a cell membrane, it will develop a force which it draws water through a semi-permeable membrane. Now, the solution from a highly concentrated solution will be lost to a highly concentrated, whereby now, if the cell was highly concentrated, we are placing a cell inside a solution that is highly concentrated. What's going to happen is that the cell will lose water to the surrounding. Instead of water moving from the beaker into the cell, now the cell is the one going to lose water into the beaker. That's through what? Osmotic what? Pressure. Is that pressure that enables what? the cell to lose water to the surrounding. So that was my time. Before I wind up, at least be able to know what is osmosis. Definition of osmosis, there are two. Number two, the type of solutions that are in osmosis. We have three types of solutions that are used in osmosis. Number one, it is isotonic, a solution that are having what? The same concentration. Number two, hypertonic, the solution that is highly concentrated. That means it has more of